right, so welcome everybody to the session on species delimitation. My name is Sarah Boyer, and I want to begin by giving a big shout out to all of my co-authors, especially Shahan Darkarbadian, who co-led this project when he was a postdoc at the MCZ at Harvard. He's now the curator of entomology at San Diego Natural History Museum. I'm at Macalester College, which is a small undergraduate-only liberal arts institution in St. Paul, Minnesota. And here you see our wonderful student team who worked with us on this project. So Rena, Phoebe, and Nathaniel performed the bench work for the work that you're going to hear about today. And Haley, who you see on the left, did all of the data analysis and also produced many of the figures that you're going to see later on in this talk. Okay, so I'm going to begin with your friendly reminder that the vast majority of species on our planet remain undescribed. My lab is engaged in the work of documenting and describing species, trying to aid in the project of shifting that proportion of undescribed in the described direction. And probably many of you in this room are engaged in similar kinds of work. And those of us who do these kinds of projects are faced with this little question: how do we identify and delimit species? That's the topic of today's session. So one of the ways of conceiving of species that's common and I think really, really useful, and that David Baum emphasized in his provocative plenary at the beginning of the meeting, is that species can be thought of as reproductive communities. So in general, gene flow happens within species and not between species. And this means that individuals who belong to the same species should be genetically more similar to each other than they are to individuals from different species. And if that's true, heritable aspects of phenotype, like morphology, like physiology, they should also be more similar between species, within species than between species. But of course, there are many, many reasons why these generalities don't always hold. And one of them is the case of cryptic species. So what you're looking at here is 4 different species. Each row is a species. You're looking at males and females in dorsal and ventral view. These 4 species are definitely distinct from one another genetically, but they're not distinct from one another morphologically. So this is a group of cryptic species which are really, really common in arthropods. So in these cases, we're forced to rely largely and sometimes even solely on genetic data to do the work of distinguishing species one from the other. Over the past generation, and especially during the last 10 years, methods for doing this have been developed and also refined. So these sorts of methods can be based on genetic distances, which is the world of DNA barcoding, or coalescence. So these methods work great in many, many systems. However, in dispersal-limited organisms that have extreme population structure related to geography, these methods can fail. These methods can fall short. In these kinds of cases, what do we do? So I'm going to be talking to you about one such case. These beautiful animals that you see here are the mite harvesters. They're arachnids, but they're not spiders. They're in the order Opiliones, which many of us refer to as harvestmen or harvesters. The most familiar of these is going to be the animals that many of us call daddy longlegs, like the one that you see here. But within Opiliones, the animals I work on are the suborder Cyphophthalmi. So this is a group of a few hundred species that are distributed all over the globe. They all look basically exactly like this. You're looking at 5 different species in 3 different genera. So this is a diverse collection of taxa, and they all basically look exactly the same. These animals are very small, just a couple millimeters long. They live in leaf litter, in soil, in caves, in rotting wood. And so as you can imagine, these are the kinds of animals that you don't notice or come across unless you're actively seeking them out. In addition, they're very poor dispersers. So most species have a geographic range that's no more than 100 kilometers in its largest dimension. And as I already mentioned, they're morphologically highly conserved. So as you might guess, this is a group with a lot of undescribed biodiversity. So in my lab, we're working on documenting and describing these animals. And right now, we are focusing our efforts on New Zealand. So that's where we're headed next. So what you see here is one of the genera that's endemic to New Zealand, Aoraki. And on the right is a recent mitochondrial DNA phylogeny for this genus. The colors of the names, which correspond to the colors on the map, represent different morphospecies. So you'll see that most of the morphospecies in this group do correspond to well-supported monophyletic groups, which is great. But if you'll spend another moment looking at this tree, you'll notice that there's a problem area. This is the Arachidenticulata species complex, and this is the topic of my presentation today. Okay, so with, getting my laser pointer. There it is, okay. All right, so within this lineage, there are two well-behaved species, which are Arachilongitarsa, which is shown here in green in the tree and also on the map, and Arachimeridialis, which is shown in pink on the tree and on the map. Everything that's shown in blue belongs to Arachidenticulata, which is a species that was described in 1948 with two subspecies. Those two subspecies are distinguished by size. The large one is called Arachidenticulata major. It's shown in light blue, and you can see it's a well-supported monophyletic group, whereas the small subspecies is shown in dark blue, and as you can see, it's a mess. So this subspecies, the small subspecies of Arachidenticulata, harbors a huge amount of genetic diversity, and you see some stats about that in the text on the left. This tree suggests that it's paraphyletic, although you'll also see there are a lot of very low support values here, so really with mitochondrial DNA only, these relationships are pretty unresolved. Previous authors have attempted species delimitation based on mitochondrial DNA in this group, both distance-based and coalescent-based approaches, and those approaches have suggested huge numbers of species. So numbers of species that, to those of us who have worked on these animals for many years in many parts of the world, really, they frankly seem unrealistic. So the bottom line is, mitochondrial DNA can't solve all of our problems, which I'm sure is not news to anyone in this room. Okay, so what can we do? We've turned to bigger data sets and novel analytical methods, and that's where we're going next. All right, so here's our newest phylogeny for this group of organisms based on UCEs. And what you see here is a very well-resolved tree. So in this case, all of the branches that have no numbers on them are supported 100%. So really, really well-resolved tree. We are so excited to see this result. What does this tree tell us about the number of taxa in this group? So this analysis retrieved the same morphologically distinctive taxa that we saw in our mitochondrial DNA set, and their names are indicated in color. And it also confirmed that Arachidenticulata denticulata, the small subspecies, is paraphyletic. There are three lineages of this taxon, none of which is sister to the other. So this suggests there's a minimum of 3 cryptic species in this group. All right, so what I'm going to do next is I'm going to simplify this phylogeny. I'm going to streamline it to like the bare bones that you need to be able to understand the rest of the talk, and I'm going to walk you through some results. So, okay. All right, so what do we know so far? So the columns that you see here represent numbers of species that are suggested by various types of data and various modes of analysis. So the column on the left is the number of morphospecies, and you can see there's 1, 2, 3, 4. Each rectangle represents a species. The column on the right represents the number of morphologically distinct monophyletic lineages. So we have, in this case, we have 6 species, with 3 of them being these cryptic lineages of Arachidenticulata. But of course, we're not going to stop with this tree. As promised, we're heading to the world of machine learning. So machine learning uses dimensionality reduction to identify latent variables that explain the structure of data sets. In the case of delimiting cryptic species, our data are our genetic sequences, and the latent variables that we're hoping to identify are the species. So we were working with SNP data that was derived from our UCE data matrix. And in order to analyze it, we took 2 different approaches. So machine learning can either be supervised or unsupervised. In unsupervised machine learning, you don't incorporate any a priori information. The algorithm merely looks for the structure of the data. In supervised machine learning, you can incorporate training data. So this is our taxon of interest within which we're hoping to delimit species. We use 2 different training data sets in our supervised machine learning. So one was drawn from animals generally, within which species boundaries are well understood. So there were big animals in this data set, small animals, vagile animals, dispersal limited animals. We also used what we're calling our custom training data set. So this was a data set that was derived from another group of dispersal-limited opiliones within which species boundaries are already well understood. Okay, so what do all of these methods tell us about the number of species in this group? So, all right, so here you see the same results figure, but I've expanded it. So as a reminder, we have 4 morphospecies, and then when we add information from phylogeny, we know that we have a minimum of 6 species. I'm gonna walk you through first our results from unsupervised machine learning. Those are the 4 columns on the right. And you can see that they generally converged on a similar answer with just one of our methods, random forest, adding 2 cryptic species. So most of these methods found a total of 8 species, with 5 of them being cryptic. One of them found a couple more. When it comes to supervised machine learning, that's what's highlighted here. So recall we analyzed our data using both a general training data set, that's shown here, and it found an answer very similar to what we saw in our unsupervised machine learning approaches. However, when we incorporated our custom training data set from a biologically very similar taxon, we found a lower number of cryptic species, 6 species total in the complex and only 3 cryptic species. And then just for good measure, we also performed species delimitation based on mitochondrial DNA, both previously published data and also new sequences that we derived from our UCE bycatch. And we performed a few of the standard kind of modes of species delimitation on this data set. And as you can see, our cryptic species were split into many, many more, which is exactly what we expected to see. All right, so given there's a large number of possible answers to the question, how many species are there within this complex? How should we proceed? Especially thinking about this, how should a taxonomist proceed? So we focused on 2 criteria. Number 1, we're interested in answers that are congruent across different data types and different modes of analysis. We're really interested in concordance. And then number 2, when it comes to systematizing species, to naming new taxa, our point of view is that one should take a conservative approach and only name new taxa when there's a huge amount of evidence that leads us to do so. So with that in mind, with those priorities, we favor the solution that's highlighted here. So a total of 6 species in this group with 3 cryptic species. So we've gone ahead and named 2 of those cryptic species. Okay, so now I want to take you through the takeaway messages from this work. So number 1, species delimitation is particularly challenging in dispersal-limited organisms that have very strong population structure. It's a problem, but supervised machine learning approaches that incorporate especially custom training data sets from biologically similar taxa can help us to overcome these problems. And finally, our point of view as taxonomists is that we favor conclusions that are congruent across different data types, different modes of analysis, and we take a conservative approach to naming new taxa. And if you're interested in this work, we very recently published a paper on it. MPE had a special issue on machine learning, and you can check out the publication led by my wonderful student Haley, who you see here posing with another one of her favorite opiliones. And with that, I want to give one more shout out to my collaborators, and I'd be happy to take a question. Yes?
Hi, thanks for that. So can you expand a little bit more on the training data sets you use? So it's also UCE data?
Yeah, so it's also SNPs that's derived from UCE data sets. So like exactly the same kind of data set that we're using, but in taxa where species boundaries are well understood.
And from your kind of your understanding, like, do you think that in general traditional taxonomy is done quite similar in these groups, or compared to the groups that you've been working on?
That's a great question. I mean, I think that all of us who are interested in counting species are faced with this problem that every species is, at least to some extent, biologically different from every other one. And I'd be happy to talk more about it later. All right, thanks everybody.
All right, I'll just get started if that's all right with everyone. Hello, everybody, and today I'm Gavin Munson from Utah State, and today I'm going to be talking about the spiny witch hazel flower galling aphid. These are galling aphids that exhibit both sexual and asexual complex life cycles that host, alternate, and specialize. So just to familiarize yourself a little bit with the ecology of these guys, this is their first host, witch hazel. On the left are the galls that the aphids are making. In the middle are the fruiting structures of that plant. On the top right corner is the flowers that witch hazel makes, and just kind of a general in situ picture, right? In the forest, right? They're kind of a shrubby bush in the understory. And currently there's 3 known North American species of witch hazel, and yeah. So, I thought I'd play a little, like, Where's Waldo game of can you spot the aphid gall, right? So, when I'm out in the field, the fruiting structures look very similar to the galls. You can kind of, I mean, now that you're familiar, they look globular and round, and so do these fruits, right? So, if we look right here, you can see the little spiny guy right there, while all the rest are the fruits. And on this one, there's actually 2 of them. There's one right here in the shadow, and there's one way up here. Kind of cute. Here's a couple more cool pictures. This guy is also hanging out in the shadow right here, and here's 2, front and center. You'll also notice on here is another aphid that does a leaf gall that's a totally separate species of aphid, actually a couple different ones that make leaf galls on the same plant. Here's a couple more close-ups. Very nice, pretty pictures. Here's a couple old galls from previous years, so you can see they kind of, after the aphids emerge and leave, those galls kind of harden and just become crusty little flaky leftovers. So kind of what happens, so let's talk about their life cycle and their migration, right? What happens if I cut open that gall, this is kind of what we see inside, and just to kind of orient yourself with their life cycle, right? So starting with the little black dot, that's the egg. The eggs are laid near the stem of the flower, and out of the egg emerges Gen 1, or the fundatrix. This is the founding mother, and what she's going to do is she's going to waddle over to the flower and start feeding on it. That's going to cause a physiological response from that plant to start forming that gall around it, right? Galls are basically just insect architecture. And then she's going to lay tons of offspring, and those are all going to be Gen 2s, these alates. And we can see a couple of these. So this founding mother is probably buried down. All of these are her children inside of this gall, so she's probably buried down underneath them. She's just a little tiny bit bigger than all the rest, and all the rest are nymphs, besides one. In the foreground here is the alate. So all these nymphs will grow up into adults and get wings, crawl out of a little exit hole at the bottom, and fly away and migrate to their sunken host, which is birch, Betula species. And on birch, they make these leaf galls. They're corrugated leaf galls. They look like kind of accordion shaped, when typically it would just be a flat leaf. And then they kind of hide up in those corrugations. So when these alates fly over to birch, this is going to happen late in this season, and they're going to have their offspring on birch, and this is going to be an overwintering coccidiform, so it's just going to kind of sit there. It's very similar to like a scale insect, just going to sit there over winter and not really do much. They have extremely reduced morphology, little tiny dinky legs. And then they're going to have their own offspring, and the following spring after they overwinter, and that's going to be the Apteris virginopores, or virginopera. So their Apteris means wingless, and virginopera, they came from parthenogenic parents, and they're going to give offspring through parthenogenesis. That's kind of what that means. But their offspring are going to be migratory sexupera. Sexupera means that they have sexual embryos. So they came from parthenogenesis, but their embryos are actually sexuals. And migratory, they're going to migrate back to witch hazel. So there's some close-ups of, there they are inside those leaf corrugations. You can kind of pull them apart. And this is more of a pseudo-gall. They're not fully enclosed like the other gall is a true traditional gall. These ones are pseudo-galls on the leaves. So yeah, that's just kind of a basic overview of their life cycle, right? The flyback to witch hazel, the two sexuals will be on witch hazel, meet up, and lay another egg to continue the cycle. So kind of complex. So I went out into the forest and collected some of these, and I also had collaborators that sent me some. I made this little distribution map, and yeah, I quickly threw together a neighbor joining tree with preliminary mitochondrial CO1 data. So since a lot of their life cycle is parthenogenic, I think mitochondria is sometimes controversial because it's only maternal, but so much of their life cycle is just asexual that I'm not sure if that matters. I'm sure there's maybe a little bit of things going on with that one sexual generation, but it's interesting. And this is what I had at my disposal. So I created this quick neighbor joining tree with 100 bootstrap lipids to get these support values, and it seems like there's these three clades kind of popping out. There's this kind of lowland clade in the green, this northern clade in the blue, and this highland clade in the red. And so to complicate the complexity of this expands further to some of their ecology, where this lowland population is only using river birch and typically found at lower elevations. That's Batuta nigra. While the highland population, this northern population, is using other birch species, which this lowland population isn't found at all. So they're kind of becoming less host specialized, or were less host specialized. Not sure really how that went down, but some of them might be like locking on a subcycle on just the birch, because in some of these northern areas there's no witch hazel present at all. So that's really interesting. I'm not sure what's going on there yet. And then this highland population in red is possibly a combination of the two, something really complex, and they're typically only found at higher elevation ranges. So I threw together this quick little table. I haven't really done any actual statistics on this yet, but just like a quick check of, oh yeah, these highland populations are kind of hitting a base minimum elevation that they're ever found at. So that's interesting. And to continue this work, I'm going to put these guys on slides and mount them, look at some of their morphology. Yeah, kind of check them out. You can get real in on some of the morphometric data, like here on the antenna, all these individual little rings, sensori, might be different between the species. I think that's how they're able to find their hosts. So I think there's something going on there. And you can also look at the embryos of the Gen 5 population, and that will give you an indication if they're going to go sexual or if they're going to go asexual. So you can look here on these little wax glands on their back are very different orientations of how they're structured along their back. And I don't know exactly why that is, but it's just a really specific morphological difference that's popping out right now. So moving forward, I'd like to continue doing more. It's very preliminary right now, so I'm not really sure, and the whole reason why I'm here is to learn more methods and things to do, right? So I'm hoping to get some nuclear DNA done. I don't think mitochondrial can answer all of my questions. So I'm looking at this bicycle gene from my collaborator David Stern at the Genelia Institute in Virginia. These bicycle genes are involved in a lot of the gall development stages. And endosymbionts are huge in aphids. They're controlling a lot of there's some papers suggesting that they co-speciate with them, so I could possibly look into that. During this conference, I've heard a lot about RADseq and UCE, so that's been super helpful to me, and I think I'd like to continue doing something with that. But I'd love to hear your ideas, if anyone has any suggestions of things that I should try. And morphometric data, I have no idea what to do with that, besides just throw it into a PCA. So if you have ideas of what to do with morphometric data, I'd love to hear it. And yeah, lots of people to thank. That's my talk. Any questions? Yeah.
Nice talk. So if you go back to your antennae close-ups, there you go. So if these are being used potentially to sniff out metabolites that are, from either one host or the other, and one of your populations any immediate morphological distances, or differences at least in that part of the body, or no?
Not yet. I haven't. It's kind of something I could continue to hope to look at. I just got trained on a new slide mounting technique that I haven't fully employed yet, so I'm going to be making a whole bunch of slides this next semester and looking at them closer. But I haven't like fully started the morphological data yet, but I'd like to get more into. Thanks. Yeah, one more.
Hello everybody, my name is Pedro. I'm going to present part of my PhD research that I did in Brazil. So I'm going to talk about how hybridization and rapid diversification can impact species delimitation in Petunia. So first, probably all of you know what a Petunia looks like, but you probably know Petunia hybrida, that it's used in gardens everywhere, but the wild species, they are divided mainly into two groups. One of them is the short corolla tube clade. As the name suggests, they have a short corolla tube. They are all purple and they look very similar. They are all bee pollinated, but I'm not going to be talking about them. If you want to know more about them, you can go to my friend's talk this afternoon at the phylogeography session. She's going to be covering several of these species. I'm going to be talking about the long corolla tube clade. So this clade's very interesting because we have some diversity in morphology and pollinators. So here we have one that it's red and hummingbird pollinated. Here we have some white hawk moth pollinated species. We also have one that it's purple and bee pollinated species. And also some that are very strange looking and either really rare or newer species. So I don't have data on them yet, so I'm not going to be talking about these two. But also, we don't only have like this diversity of the known species, but we also have like several contact zones and potentially hybrid populations. So here we can see this one that kind of looks like an intermediate morphology between two different species that are pollinated by different organisms. And here you can see that like a different population that you have in the same population, some individuals that look more like one parent and other ones that are more like in the intermediate morphology. And also some other ones that could be hybrids or could just be maybe a new species. So because all of this, our goals was to create a new phylogeny with several of these lineages belonging to the long corolla tube. Also evaluate the status of some potential new species and also see how sampling at mixed individuals could impact species delimitation in the group. So to do this, we collected more than 300 individuals spanning several known lineages and also some putative new species and contact zones. I'm not going to go over all of them, but I just wanted to show you some of the diversity. So here we have Petunia parodii that is distributed in Argentina and Uruguay and Brazil. They are also distributed in different biomes. Then we have Petunia secreta, that it's a rare and really endemic lineage that occurs only like in a very small region in southern Brazil. We have these other strange looking atypical populations, maybe hybrids. So some of them are very similar to Petunia secreta, some of them are more of the intermediate morphology, some of them are intermediate morphology between different parent species, so this one could be like a hybrid between a white one or secreta and the exerta, that it's this one. So you can see it has the same exerted stamens as Petunia exerta. Exerta is also really rare and endemic in this region in southern Brazil, and it occurs in a very similar habitat as Petunia secreta. And we also have one that is one species that it's widely distributed. So it occurs in Argentina, Uruguay, and Brazil. and it's hawk moth pollinated. Okay, so I'm gonna go over the main results that we found. So regarding Petunia parodiae, we can see it is divided mainly in the biomes it occurs. So here in the dark green, we have the species that are in the Chaco biome, and light green, we have the populations that are distributed in the Pampa grassland biome. But what is also interesting is that we have some contact zones between Petunia parodiae and Petunia axillarius, that it's the other white hawk moth pollinated species. And populations from these contact zones, they fall within either Petunia parodiae or sometimes within Petunia axillarius, suggesting that these populations could be hybrids between these two different species. The other thing that we found that was really interesting to me is that Petunia axillarius, that it's the white hawk moth pollinated species too, is paraphyletic. So we can see all in these shades of blue, we have one that corresponds to the south distribution of the species. Here in this blue here and this blue here, we have the central distribution of the species. And here in the dark blue, we have the north distribution of the species. But it's interesting that it follows the budding speciation pattern, where we have one species that it's very widely distributed and very common, that gives rise to new endemic and rare species. So like this one, that's Petunia secreta and Petunia excerta. Potentially, we are not sure about the position of Petunia excerta here on this tree. But what is also interesting is that they have a very, very weak reproductive barrier, so we can find hybrids between Petunia axillaris and Petunia excerta, so the white one and the red one, and gives rise to something similar like this. And these populations are actually very common, and we can find them every year that we go to the field. They are always there. And there are also some other hybrids that are a little more rare, like this one, that it's a hybrid between Petunia secreta and Petunia excerta. And now going to the species delimitation part of my talk, that is probably that's bringing you here. So we tested different methods, and we were interested to see how sampling this at mixed individuals could potentially impact how we delimit species. So for this, we tried different scenarios. So one scenario here, using BFD star, we pruned all of our contact zones and hybrid individuals, and we tested models ranging from like the current taxonomy to one where we split between axillarius into different lineages depending on the geographic distribution, to one that it's an oversplitting scenario where each sampled lineage corresponds to a new, each sampled population corresponds to a new lineage. And we did that to the data set where we left our admixed populations. And I'm not going to go over all of the results, but the main message here is that testing different data sets where we varied the individual sampled based on the most admixed, the least admixed, or random individuals. These results were very conflicting. So here in scenario 2, where we didn't leave any of the admixed individuals, we found that depending on which data set we used, we would get a different result. But the model 2D, where each population was its own lineage, was the best one, and it was selected in 2 out of the 5 data sets that we tested. And when we sampled the individuals that were admixed, the model 1D, where each lineage which population was its own lineage, was the best one in 3 out of the 5 data sets. And it's a little intriguing because we would expect that collecting at mixed individuals, we would get less species than more species. And the other method that we used was delineate. So for this scenario, we don't use the SNPs. We use a tree as our input. So this is interesting because we can tell the software which are the lineages that we know that belong to that species. So here in the blue branches, we have the ones that we know that belong to those species. And the yellow branches, we allow it to vary depending on the scenario that delineate suggests. So what we found is that some of these contact zones, as expected, they sometimes fall within one parent and sometimes with the other parent. So here, contact zone 3, we have some populations that fall within Petunia axillaris, some fall with Petunia parodii, and the same here with CZ1 that falls within Exerda or axillaris. What's also interesting is that potentially new species that we collected, they are more similar to what a Petunia secreta would look like. They were assigned to Petunia axillaris, and we believe that could be due to the Petunia axillaris paraphyletic status. But it's interesting to see here that the lineage suggested a more conservative scenario than BFD star and placed the unknown lineages within an already known species. But overall, what I wanted you to get from my talk is that Petunia axillaris is paraphyletic and it may encompass more than one taxon. It follows the budding speciation pattern where one widely distributed species gives rise to rare and new lineages. Also, that we can get conflicting results in the limiting species because some methods are more conservative while others are better at capturing population structure rather than species themselves. And that it's a very complex task to delimit species in cases of rapid radiations and where we can find frequent hybridization. This is a case in Petunia, but it's probably common in several taxa. And the overall conclusion is that there is no magical method for us to delimit species. We have to rely on the old integrative taxonomy and use morphology, genomic, ecological data to try to get the best species delimitation that we can get with the data that we have available at that time. And with that, I would like to thank all my amazing co-authors, all the funding agencies, and all of you for being here on the last day. Yeah. Right on time. Yeah.
So do you know anything about moiety levels in these? Are they all the same?
They are all the same. They all have the same chromosome number, as far as we know.
Okay.
Yeah.